Hello everyone, this is Chandrasekhar from Chandu Biology classes. Finally, the JL and DL notification has been out. And we know that in JL we are having very limited post. That is one post in both botany and zoology. But in DL we are having 19 posts in botany as well as the zoology also. In this particular session, I will do the complete analysis of the syllabus and I will help you with complete strategic plan. And I will explain you, from every single unit, how many number of questions that we can expect in the exam. What are the most important topics that you have to focus on while doing your preparation, and each chapter, how many marks that you can get, and what kind of questions that you can expect in the exam, what kind of books to be followed. I will explain you everything very briefly and elaboratively. It is a really informative session and extremely useful session to all of you. And those who are really dedicated to crack the exam in the field of zoology, I'm requesting everyone to watch the video till the end. Right. Before that, the new batch is commencing from the 11th January onwards. Okay. In this, the batch will go on up to 100 days, where you can get complete live and recorded sessions. You will get well-crafted PPT, and you will get complete hard copy of the study material, which are nearly 10 books, and you will get chapter-wise test and 10 mock test. Okay. Those who are really dedicated and passionate to crack the this DL program, please join the classes. You can take up to 7 days of absolutely free classes, which is starting from the 7th January. For further details, please download our app. Okay, now I hope everyone gone through the notification and everyone has gone through the syllabus also. So again, everyone look into this. If you look into the syllabus, there are, first unit is called general concepts, second unit is card date, non-card date, third unit is card date, cell biology. Now you look into the cell biology. It is the combination of cell bio also, molecular biology also, as well as rDNA technology also. It's a kind of a very big unit. Then followed by another big unit, genetics. And in the entire your syllabus, there are two big units are there, very big units. One is the human physiology and the next one is the immunology. Anyway, evolution, developmental biology, histology, ecology. When compared to the human physiology and immunology, these are little smaller units. Okay, so friends, total how many units we have? 11 units we have. How many number of questions that we can get in the core paper? You know very well, we are having 150 questions that we can get in the core paper. So what I can tell you, from every single unit, every one of you listen my words, from every single unit, minimum 11 to 13, or in some units you may get up to 14 questions also. I am telling minimum. Hence, it is very important, friends. You have to focus on every single unit without fail. Okay. Now I will do the complete analysis of cell biogenetics, human physiology, evolution, developmental biology, ecology, and immunology. So in other session, madam will do the discussion of the histology, general concepts, non-coordinate, and coordinate. Okay. Now let's focus. Now let's start with the biochemistry. Now, friends, if you look into the biochemistry, especially in the human physiology chapter only, they have included the biochemistry, is it? But for the zoology people, the biochemistry syllabus was very less. But if you look into the botany people in the DL, you are having a vast syllabus in the biochemistry also. Anyway, how many number of questions that you can expect, friends? You can expect minimum 3 and you can expect maximum 5 in the biochemistry. Why? Because biochemistry is actually included in the human physiology as a small subtopic. And I know it's very difficult for all of you not to prepare Leninger, White and White or any other book. But you don't need to worry. I will provide you the excellent expert designed study material, which will cover A to Z of the entire syllabus. Right. Now, if you look into the biochemistry, you have to majorly focus on glycolysis, Krebs cycle, electron transport chain, oxidative phosphorylation. Correct, na? So when you are reading the glycolysis Krebs cycle etc, especially in the glycolysis and Krebs cycle, most of the students will just read 10 steps of the glycolysis and all the steps of the Krebs cycle. You never get the questions from the steps of the glycolysis, steps of the Krebs cycle. Most of the time, in order to make the question very difficult, they will raise the question especially from regulation. How the glycolysis will be regulated, what are the regulatory enzymes, how they are going to regulate it, how the Krebs cycle is going to regulate it, how many number of ATP, how many number of NADPH, how many number of the FADH2, how many number of the GTP is getting produced per one glucose molecule or per one pyruvate molecule in the entire cellular respiration or in the entire Krebs cycle. So there are different ways of asking a question. Apart from this, the most important one is most of the students will neglect also. That is, they will read the electron transport chain and they will read, how many electron acceptor complexes that are present on the inner mitochondrial membrane. They will read everything. But most of the students will skip completely the inhibitors that blocking the electron transport chain. So which inhibitor is blocking the first electron transport system, which is blocking the second one, which is blocking the third one, which is blocking the fourth one, which is blocking the F0 F1 complex, that is the ATP synthase enzyme. So these kind of questions they will ask, more times. Anyway, we will discuss very elaboratively in the classroom. And they have mentioned a very, limited amount of the chemistry here. Whereas amino acid chemistry, when you're reading the amino acid, most of the students, again, who are preparing for the JL and DL, they will just do the they will just read the classification and they will leave it. No, it's not like a classification. You're not writing, 10th class or 11th standard exam. If you just read the classification, there's no use. You have to focus on the pKa related numericals. You have to focus on the PI related numericals. You have to focus on each and everything, because the numerical part is the little difficult and little depth you have to read. Then followed by, you have to read the proteins. Anyway, they have very clearly mentioned primary, secondary, tertiary and quaternary structure of the protein. Again, we can expect the questions, friends, what are the major stabilizing interactions that are found in the secondary structure, major stabilizing interactions in the tertiary structure, quaternary structure. And especially when you're reading the secondary structure, they will again ask the questions from hydrogen bonding in the alpha helix, hydrogen bonding in the beta pleated sheets, and how many number of amino acids are present in one helix formation, how many number of amino acids are present in the one beta pleated sheet formation. These kind of questions also they will ask very commonly in the exam. Then chemistry of carbohydrates, it is very easy topic. Question also comes for very short, but the only point is that you have to remember maximum amount of the information. You have to focus on classification of disaccharide. An example, the major linkages between the some of the disaccharides, such as sucrose, maltose, lactose, and very important polysaccharides such as inulin, starch, glycogen, cellulose, cellobiose. There are many are there, right? Proteoglycans, heparin, chondroitine sulfite. So there are multiple polysaccharides are there. We have to read all their linkages. What is their major glycosidic linkage? So these are all the type of questions that we can expect in the exam. Beyond this, there is no way of asking the question. Then the next one, cell biology. I told you, right? In your zoology syllabus, they have included cell biology, molecular biology, and rDNA technology in a single unit. But trust me, this unit will be the biggest unit in your entire syllabus, along with the human physiology. Now, when you are reading the cell biology, the standard reference books are either Karp or Ladish. You can read as the standard reference book. There's no doubt about it. Now, in the cell biology, what kind of questions you will get? You will get the questions from the organelles. You will get the questions from the chromatin organization. You will get the questions from cell cycle regulation, and you will get the questions from cell division. Most of the people who are preparing for the JL and DL, first, let me talk about the cell division. They will just read the stages of the mitosis, the stages of the meiosis, and the events that are happening in the mitosis, events that are happening in the meiosis. No, that is not at all sufficient. So, even 11th standard students also know the complete stages of the mitosis and meiosis. Then what is the difference between you and the people who are preparing for the NEET? Here, most of the questions will comes from the regulation part, which means, so if you look into the cell cycle, we are having a multiple phases: G1 phase, S phase, G2 phase, and M phase. You all people read extremely, well in many years. There are many type of CDKs. There are many type of cyclins are actually involved in regulating every phase of the cell cycle. You people commonly know about it. So you can expect most of the questions from here. What kind of cyclins are regulating, what kind of CDKs are regulating. And very important, friends, they will link the cell cycle with the cancer biology and they will raise the questions. So it's very important that I want you to become super experts in the cell cycle regulation concept, which is extremely important. I can say that there is no question paper without the question from the cell cycle regulation. That much important it is. At the same time, you have to focus on structure of heterochromatin, euchromatin, and transposon. Actually, in your zoology syllabus, this heterochromatin, euchromatin, which is the chromatin organization, that they actually mentioned in the chapter of genetics actually. They mentioned in the chapter of genetics actually, but here only we have to read, okay, because it is very easy. Here, in the TSPSC DL question paper also, we got two questions from this chromatin organization. In the heterochromatin, what kind of proteins are involved? An example, if you take a H3 and H4 histone proteins. In the euchromatin, what kind of modifications H3, H4 undergoes? Whether they are undergoing methylation, whether they are undergoing acetylation, whether they are undergoing phosphorylation. Same with the heterochromatin also. So these kind of tricky questions that we can expect in the exam. And again, they will raise the question about telomere, centromere. And when you look into the transposons, very, very important, friends. Zoology people also has to understand the type of transposable elements that are present in the animals, humans. Even you should have a general idea about the transposons present in the plants also, right? And what is the structure of the transposable element, what is its composition, how the transposable element can jump from one area to the another area, how the transposable elements are getting regulated in our body, their regulatory mechanisms especially. We have to focus on everything. And in the entire cell biology, friends, which is the most easiest topic, undoubtedly the cell organelles. There's no doubt about it, because if you are getting a question from the cell organelle, there are only three ways of asking the question. What is that? One is from the structure, another one is from the function. A very, 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 very important, again, most of the aspirants will miss it. The enzymes involved in every single cell organelle. Then the deficiency diseases related to that specific organelle. In the TSPSC DL also, most of the people forgot, studying deficiency diseases that are related to the cell organelle. But surprisingly, they raised a matching question from various organelles, the enzymes associated with it, and the deficiency diseases that occurs because of the deficiency of that specific enzyme, like Tay-Sachs disease, Gachers disease. There are different type of diseases are there. So that kind of questions also we can expect in the exam. And if you ask me, sir, should we read all the cell organelles? Undoubtedly, my friends, you have to read every single cell organelle. There's no doubt about it. But I will give you the preference. I will tell you the importance, how you have to read, or the sequence of the organelles that you have to focus on. Give the highest importance to the plasma membrane. When students are reading the plasma membrane, they just read structure of plasma membrane, function of plasma membrane, they will leave it. No, it's not like that. You people has to read extreme depth of the plasma membrane. the type of carbohydrates present, their function, the type of proteins present, their function, the type of lipids present, their function, how the transport mechanisms are happening, like what kind of substances can be transported, what kind of substances cannot be transported across the plasma membrane, all osmosis, diffusion, facilitated diffusion, phagocytosis, endocytosis, pinocytosis, everything you have to read extremely depth when you are reading the plasma membrane. Anyway, I will explain to all of you very depth. After plasma membrane, remember the nucleus. Then the third one will be the mitochondria. Then the fourth one, RER, both the RER, SCR, especially the protein sorting in the RER and SCR. Then friends, followed by the Golgi complex, then followed by the cytoskeleton, then lysosomes and peroxisome. So friends, whenever you are reading the cell organelles in the cell biology chapter, it is extremely important you people has to focus on all these 8 organelles from A to Z, from scratch to advanced level. From very basic to the very advanced level, you people has to read completely. Very extreme depth you have to read. Then only you can answer the given questions. Anyway, I will teach all of you in such a way that you get a wonderful knowledge on the given concept. There's no need to worry about it. Then the next one, molecular biology. I don't know, some of the, or not some of the, most of the zoology aspirants, I really don't know, they feel very, confusing and difficulty with the chapter of molecular biology. But friends, molecular biology is a kind of a chapter which is highly weightage also, kind of a difficult unit also, and kind of a unit which is completely conceptual. Very limited amount of the topics that you have to remember. Most of the topics are highly conceptual. So you people should have a wonderful concept. If you have a concept, trust me, 90% of the questions that you can answer so easily in this particular unit. And trust me, I'll teach every single unit in such a way that you will get a wonderful idea on every single minute concept. It's my guarantee for all of you. Now, anyway, what we have to read in the molecular biology, we have to read the 3 fundamental processes. An example, replication, transcription, translation. We have to read all these. Replication in the prokaryotes also and eukaryotes also. Transcription in the prokaryotes and eukaryotes and enzymes associated with it. Translation in the prokaryotes and eukaryotes. Then followed by, friends, gene expression in prokaryotes and eukaryotes. These 4 processes are common. What are those? Replication, transcription, translation, and gene expression. In the gene expression, again, most of the students, what they will do, they will just read the operons. That is the prokaryotic gene regulation, and they will leave the eukaryotic gene regulation. Friends, eukaryotic gene regulation is also extremely important for the exam. You have to focus on, because prokaryotic organism, there are only 1 or 2 levels of gene regulation occur. In case of a eukaryotic organism, especially human beings, multiple levels of the gene regulation will occur. That we have to read extreme depth. Then only we can cover that. And especially, we also get a questions from post-transcriptional modification, which is only happens in the eukaryotic organism. So what exactly I'm talking is, I'm talking about the 5' capping, and I'm talking about 3' polyadenylation, and I'm talking about this splicing. Again, most of the students, splicing means they just read the definition of the splicing and mechanism of the splicing. No, splicing means you have to read extreme depth. You have to read varieties of splicing. You have to read the self-splicing. You have to read the spliceosomal mediated splicing. You have to read the alternative splicing. You have to read the trans splicing and their mechanisms, and what kind of introns are present in the mitochondria, what kind of introns are present in the nucleus, how they will be removed out, what is their major mechanisms. Extreme depth we have to read. Now, whenever you are reading the replication, you should focus on, remember, you should focus on the rules involved in the replication, enzymes involved in the replication. Very, very, very important, the process of the DNA replication. Same with the transcription, the enzyme, their structure, the process. And when you are reading the translation, you should have a very good idea how the mRNA is getting ready, how the tRNA is getting ready, how the rRNA is getting ready, how all these three are getting assembled for the synthesis of a protein. And we have to read extreme depth. I can't explain the molecular biology in single slide to all of you. It's a so depth concept, but I'll teach you, very elaboratively. You get a wonderful idea on the concept. Apart from these three common fundamental processes, we should also focus on DNA repair mechanisms also. DNA repair mechanisms are also extremely important for our exam. We should not skip them under any circumstances. Right. We have to focus on every single DNA repair mechanism, such as we have to focus on the mismatch repair. We have to focus on the nucleotide excision repair. We have to focus on base excision repair. We have to focus on direct repair or the photoreactivation. We have to focus on homologous recombination. We have to focus on double strand breakage repair. And there are multiple pathways. All the enzymes involved in the DNA repair mechanisms, everything we have to read. Anyway, I'll explain you extreme depth. For this, I encourage you to go through the Watson. But if you feel extremely difficulty to read the Watson, anyway, I'll provide you high quality study material. You can easily, understand from that. Then immunology. Now, if you ask me one question, sir, can you tell me an important topic in the immunology? No, I can't tell. Why I can't tell a very important topic in the immunology? What is the reason? One of the reason is, friends, the immunology chapter works in a cascade way. What is the meaning of a cascade way here? If you don't understand one topic, if you don't have a conceptual knowledge on the one topic, trust my words, you can't understand the second topic. Without understanding the second topic, you can't understand the third topic. If without reading first topic, second topic, and third topic, if you just enter it into the sixth topic directly, trust me, your knowledge on the chapter will be zero. There's no point of reading also. That's why in case of immunology, my friends, it is very important to read the entire unit. So how to read this unit? Again, most of the zoology students, along with the molecular biology, they feel very difficult in the immunology also. Then how to read this immunology? Can I read it very easily? Yes, of course you can read it very easily. There's no point of worried about it. It is, I can tell you, 95% of the immunology, 95% I'm telling you, my friends, it is conceptual. Only 5% of the concept to be remembered. That's all. So if you have an extraordinary knowledge on the concept or the subject, you can qualify, you can answer any kind of a question. So what to read? How to start the immunology? Whenever you are reading the immunology, first, you have to read the cells of the immune system. What, friends? Cells. Like, what are the major cells that are involved in the immune system? You all people commonly know that all the type of the WBC will be involved in the immune system. So majorly we are studying the lymphocytes, and we are studying the non-lymphocytic cell also. In simple words, granulocytes and agranulocytes. So in the lymphocytes, we should majorly focus on the B lymphocyte, T lymphocyte. When you are reading the B lymphocyte, you have to read plasma cells, memory cells, and extreme depth and their development. T lymphocyte, we have to focus on T helper cell, T cytotoxic cell, T suppressor cell. See, friends, whatever I'm telling you in this particular slide is a very small. I can tell you immunology is like an ocean. It will take for me 1 month to teach immunology only for all of you. That much depth we have to read, my friends. Then when we are apart from the lymphocyte, we should give a focus on monocytes, macrophages, differences between monocytes and macrophages, mast cells, basophils, eosinophils, and neutrophils. The surface markers associated with it, the structure of them, functions of them, and mechanism of them. These are all what? Just cells, only cells. after studying the cells, because once you have a good knowledge on the cells of the immune system, then it is very important to understand the antigens. Very important. Most of the students, when they are reading the antigens, they don't understand what exactly to read on the antigen. You have to understand the difference between the hapten, the difference between the adjuvant, which is more effective, and what is the difference between the epitope and paratope. Everyone know that very well. I can't explain you extreme depth here itself. Paratope will be the part of the antibody and epitope will be the part of the antigen, which is the epitope is the most biologically active part of the antigen, which cause the pathogenicity. And what type of pathogenic organisms release what type of the epitopes? How our immune system will recognize those epitopes? In simple words, what kind of receptors that can be used by these particular epitopes? What kind of receptors are present in our immune system? Which example? An example I'm talking. We have a toll-like receptor. So different type of a toll-like receptors are recognizing different type of epitope. So what are the type of toll-like receptor also we can expect in the exam? Now, after reading the antigens, we should give the highest importance to the antibodies. Again, most of the students, when I tell the antibodies, they will just remember structure of the antibody and they will just remember functions of the antibody. No, friends, that is not sufficient. You are not preparing for, intermediate level. You are preparing up to PG level. You should know the enzymatic cleavages of an antibody. So what enzyme will cleave which part of the antibody? An example, mercaptoethanol, how it will cleave the antibody? Papain, how it cleave the antibody? Pepsin, how it will cleave the antibody? That's a very important you have to focus on. Then friends, a very, 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 very important topics, which we call it as MHC, major histocompatibility complex. So there is a type 1, type 2, type 3. Anyway, we know that type 3 is not actually related with the type 1 and type 2. You know that very well. So they will majorly raise the questions from type 1, type 2, the structural differences between type 1 and type 2, functional differences, how many number of amino acid that can present on the type 1 MHC, how many number of amino acid containing antigen that can be situated on the type 2 MHC. Functions of the type 1 MHC, functions of the type 2 MHC, which will do the endocytic pathway, which will do the phagocytic pathway, or which will do the exocytic pathway. A very common questions. Then followed by the complement system. We have a different type of a complement system: classical complement, non-classical complement. We have a lectin mediated complement pathway. We have to focus on every single complement pathway without fail. Right. Now, very, 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 very important. They specially mentioned in your syllabus antigen antibody interactions. Affinity, avidity, cross reactivity, precipitation reaction, and agglutination reactions, and ELISA. Friends, from this antigen antibody reaction, there is a possibility that you can expect the graphical questions. Exclusively, you will get the question from Scatcherd equation, which is the graphical. There is a possibility that even you can get the experimental based question also. You should read that very clearly. And the last and final one, disorders. Disorders are very easy. You don't get any difficult question because they have very clearly mentioned also, and that mentioned diseases only we will read we no need to read their pathophysiology. We have to focus on how that disease will cause, and what is the mechanism. Means that disease is affecting which system or which receptor or which cells, which surface markers. From those only we will get the direct question. Now again, I'm telling you, just to teach all of you immunology, it will take 1 year, 1 month for me. That much depth we all people has to read. Anyway, we will focus on that. We will work extremely hard with respect to the immunology and every chapter. Don't worry about it. You people can't read the entire QB. You can't read the entire Abbas. You can't read the entire Royet. Don't worry, I'll provide you the high quality material and PowerPoint presentations which will help you to crack the exam. Then developmental biology. Again, what zoology people will do, when they are reading the developmental biology, they just to focus on gametogenesis and fertilization, and they will leave the entire developmental biology. No, it's not like that. Gametogenesis and fertilization is very, very, very common. And again, some students, when they are reading the gametogenesis and fertilization, they just read in general. Developmental biology is not general, my friends. Developmental biology is specific. What exactly we have to read in the developmental biology? If you ask me, we have to focus on gametogenesis, fertilization, cleavage, nothing but blastula formation, gastrula formation, organogenesis. So when you are reading all these events, you have to read very specifically. An example, how does the fertilization will occur in the sea urchin? How does the fertilization takes place in the frog? How does the fertilization takes place in the mammals? So we have to read by giving specifications to the different organisms. That is what we call developmental biology. When you are reading a specific process, you have to take different organisms as the models, and we have to focus on. Suppose example, if you are reading the cleavage, again same. What kind of egg is present in the sea urchin and what is the pattern of cleavage? What kind of eggs are present in the insect and their pattern of cleavage? Mammals, their pattern of cleavage. Amphibians, their pattern of cleavage. Very depth we have to read. And when you are reading the gastrulation, where in the entire chapter, gastrulation is the very difficult topic. Why? Because you have to read very depth. Gastrulation is what? Formation of ectoderm, endoderm, mesoderm. This is easy. But you have to read how the gastrulation is happening in the amphibians, in the frog, in the mammals. Right? When you are reading a gastrulation in all the organisms, you have to read extreme depth of genes involved. Suppose example, you are reading the vulva formation in C. elegans. You are reading the axis formation in the sea urchin. Suppose example, if you are reading the chick development, if you are reading the placenta, types of placenta, so you have to read especially the genes involved over there, proteins involved over there. What happens when the mutation occur? Which genes are responsible for what? How many number of genes are present? Where they are located? These kind of specific questions can be expected from the chapter of developmental biology. No point of getting confused, friends. And for this, the groundbreaking and the standard reference book is the Gilbert. I know it will be a little difficult for all of you, but you have to read it compulsory. Okay. Then one of the very, very, very biggest unit in your entire syllabus is the human physiology. Why the human physiology is a very important? As I told to all the students, friends, as I told you, all of you, human physiology is the chapter which is linked with the biochemistry also. And it is one of the vast unit. What is the reason why I am calling it as the vast unit? What is the reason? Because the syllabus contain multiple units. So from every single unit, you will expect the question. Let's start with the endocrine. You will get the question. Respiratory system, you will get the question. Nervous system, blood circulation, cardiovascular system, and excretory system, thermoregulation, digestive system. They don't leave anything. Muscular system. They will raise the question from every corner. Again, if you ask me a question, sir, in the human physiology and in the immunology, is there any chapter, is there any topic called as important topic? No. Immunology and human physiology, every student has to read 100%. Not even 90 also. Not even 90% also. You have to read 100%. Then only you can. But you no need to worry. We will teach extreme depth to all of you. You can understand any concept very easily. So, majorly, let's start with the endocrine system. When you start with the endocrine system, Majorly, we have to focus on, remember all the students. Anyway, I will do the, once again, chapter-wise analysis also separately, don't worry. Endocrine system, name of the hormone, from which gland it is released, functions, and which amino acid precursor is involved in the biosynthesis of it, and their regulatory mechanism. Then finally, diseases related to it. My friends, apart from this, there is no point of getting the question. What are those? Name of the hormone, from which gland it is releasing, functions, which amino acid precursor is involved, their regulatory mechanisms, and the diseases associated with it. Apart from that, there's no other way. Respiratory system, one of the most important system, I can say that. Because why respiratory system is so important is variety of questions you can get. You can get the questions graphical. See, respiratory system and nervous system, these are all the two type of systems, my friends, where you people can get the graphical based questions. No point of getting confused over there. That's also very, very, very important. Now, especially graphical question, oxygen hemoglobin dissociation curve only. Means what happens when the partial pressure of carbon dioxide increases? What happens when the concentration of 2,3 bisphosphoglycerate got increased? What happens when the pH got increased? Right? This kind of questions they will ask. And they will also ask the questions, the relationship between the hemoglobin and myoglobin also, they will rise. Apart from this, they will raise the questions from lung capacity and different types of breathing patterns, such as tidal volume, inspiratory reserve volume, expiratory reserve volume, inhalation capacity, exhalation capacity, forceful exhalation, forceful inhalation. These kind of breathing patterns are very common. And when you look into the cardiovascular system, see, cardiovascular system and blood circulation is actually different. If you look into the blood circulation, majorly you can expect the questions from blood groups. You can expect the questions from blood clotting. Apart from this, in the cardiovascular system, they don't ask you, the scheme of the blood flow. They don't ask those. They majorly focus on, cardiac output, ECG, what does the PQR waves are representing, what is the duration, and because of the imbalance or the fluctuation in which wave, what it is actually representing. An example, ventricular repolarization represented by which wave, atricular depolarization represented by which wave, I mean to say cardiac arrhythmia represented by which wave, cardiac ischemia represented by which wave, low supply of oxygen influenced by which wave, hyperkalemia which influenced by which wave. This kind of questions they will rise from the cardiovascular system. And when you look into the nervous system, resting membrane potential and action potential, again I am telling you, my friends, here you can get the graphical question. And another important is list of neurotransmitters and their function. Which neurotransmitters comes under the biogenic amines, right? Which neurotransmitters are catecholamines, which neurotransmitters are amino acid derivatives, which neurotransmitter act like both excitatory or inhibitory, both excitatory and inhibitory, which neurotransmitters are using the metabotropic receptors, which neurotransmitters are using ionotropic receptor. These kind of questions we can expect in the matching. Excretory system, my friends, again there is a possibility of getting matching questions only. An example where the amino acids are getting secreted, where the drugs can be secreted, where maximum amount of the water is getting reabsorbed in the nephron. An example, PCT, right? Where the toxic substances can be excreted. That kind of question, where the sodium glucose symporters are present, where the sodium potassium chlorine symporters are present, where the antiporters are present, and where the water can be reabsorbed more, where the potassium can reabsorbed more. What is chloride shift? What is the reverse chloride shift? These kind of questions we can expect in the matching again in the excretory system. Digestive system, trust me, you never get any kind of a difficult question. You get a very straightforward question, but with a little twisted. I can say that digestive system is the type of a system which is memory based. the type of enzymes involved, example, brush border enzymes, where they are present, which layer secretes what kind of enzyme. That is also important. HCl is secreted from where, right? What the parietal cells are actually secreting, right? And from where the hormones are getting secreted, like a cholecystokinin, enterogastrin, right? Enterokinase, where they are getting secreted, and what is their functions. These kind of generalized questions only that we can expect here in the digestive system. But remaining all the systems, we have to read extreme depth, my friends. Then the next unit, genetics. I don't know why. Again, most of the people will feel very difficulty with the genetics. But trust me, I can tell you all the students, genetics is the chapter which is 100% conceptual. What, my friends? Genetics is the type of a chapter which is 100% conceptual. Why 100% conceptual? Because it is all analytical questions, all analytical, all logical, all numerical, all case study based, all are disease related, all are subjective. I'm sorry, subjective in the sense all are conceptual. I'm saying you have to focus on common topics such as Mendel principles, extra Mendel principle. I am not focusing on the Mendel and extra Mendel. You started reading from the 11th standard. You people, the zoology people, give the highest importance to the bacterial genetics. That is methods of conjugation, transformation, transduction. You have to focus on polygenetic inheritance, QTL mapping, type of recombination. A very, very, very important, friends, linkage. I forgot to mention it. Linkage and mapping. Then very important, you get the question damn sure. That is the chromosomal alteration, deletion, duplication, inversion, translocation, ploidy, polyploidy, aneuploidy, hexaploidy, triploidy, tetraploidy, and the diseases related. And your luck is that in the botany, they have not mentioned the specific diseases actually. But in the zoology, they mentioned specific diseases. Down syndrome, trisomy, Turner syndrome, Klinefelter's, hermaphroditism. And we will read some more diseases. Since if you focus, since they have mentioned specifically, friends, if you focus on these diseases, that is 100% sufficient. I will tell you how to read, extreme depth of this. And in the genetics also, I'm telling to all the students, we have to read 100%. We don't leave anything in the concept of genetics also, right? And extra chromosomal inheritance, where we will focus on the mitochondrial inheritance and the chloroplast inheritance. It seems to be a small topic, but extremely important topic. We will get the questions for sure. There's no need to confuse about it. Now, ecology, some zoology people, I don't know why they will neglect the ecology. You should not neglect the ecology. Ecology is also one of the most important unit, my friends. You have to read it. And another disadvantage with the ecology is it's a kind of a rigid topic. I can tell for your DL and JL, if it is assistant professor and CSR, NET or SET exam, you may get a kind of experimental based question. But trust me, for your JL and DL, I can tell you, you get a very rigid questions, which are memory based. Conceptual based questions are very less. Happily, you can read. So ecology also, my friends, we have to read 100%. I don't, tell any one of you, please leave that topic and leave this topic. In ecology, I'm summarizing it in a simple way. We have to start from organism. When we are starting about organism, majorly we have to focus on habitat and niche. So here, what we have to focus on? We have to focus on the gas principle or the competitive exclusion principle. That is a single organism. And you know very well, the group of organisms are said to be what? A population. So we have to focus on complete population ecology. The factors influencing the population, such as size, density, pattern of dispersion, survivalship curve, population growth rate, R and K selections, every parameter we have to focus extreme depth. Extreme means extreme depth. That I will explain you, don't worry at all. Type of community interaction, symbiosis, commensalism, emensolism, parasitism, competition, predation. Zoology people, whenever I'm explaining all these relationships, you only focus on You only focus on animal related example. Whereas botany people also has to read this. You people remember plant related example. But when I'm explaining to all of you, I will explain both plant and animal related examples. Don't worry at all. And group of communities, what do we call? Ecosystem. So this is one of the biggest subunit where we have to read pyramids, food chain, food web. Very, 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 very important where you get a difficult type of questions, or productivity. Grass productivity, net productivity, grass primary, net primary, grass secondary, net secondary, assimilation efficiency, digestion efficiency, ecological efficiency, consumption efficiency, all the type of questions that we can expect here. Then followed by pollution, where you people give the highest importance to the air pollution and water pollution. In that also give the highest importance to the acid rain, greenhouse effect, measurement of the BOD and COD, and the water level, the pollution level based on the BOD measurements. That kind of questions also we will get. And very rigid topic, without this also there is a no question paper, is conservation biology. It is also super important, friends. We should not neglect under any circumstances. List of national parks, the animals protected over there, their conservation status. Sanctuaries, biosphere reserves, Project Tiger Zones. They will exclusively focus on the nationwide also, and they will specifically focus on our Andhra Pradesh also. Since we are writing the Public Service Commission of Andhra Pradesh. But anyway, we have to focus on both the nationwide also and Andhra also. Then I'll explain you extreme depth. You no need to worry about it. Now, next one, evolution. Again, some students will ask me, sir, evolution is really important. Of course, my friends, evolution, I can tell you the simple words. You will get one question damn sure from theories of evolution, Lamarckism and Darwinism. Very surely you will get one question from the experiments, Oparin, Halden, Miller experiment. Undoubtedly, you get one question from geological time scale. Now, the most important and difficult topics for the people, for you people, are population genetics, where you have to give the highest importance to Hardy-Weinberg equilibrium. Here, some students will get confused. Hardy-Weinberg equilibrium, gene pool, allelic frequency, genetic frequency, Hardy-Weinberg equilibrium, their assumptions, numericals related with it, not only theory. Followed by evolution of human also, you get a one question. And very common such as convergent, divergent evolution, homologous, analogous organs, co-evolution, adaptive radiation. Again, you can expect the questions in the form of a matching also, in the form of a examples also. It is not that much difficult topic. I'll finish maximum in 7 to 8 days. We will read extreme depth. So friends, that is the best analysis of your entire syllabus. So madam is going to do the complete analysis of non-chordates, chordates, histology, as well as the general concepts. And again, I'm telling to all of you, the new batch is actually commencing from the 11th January. So 11th January to 18th January, everyone can take the free classes. If you want to, attend the free classes, please download our app Chandu Biology Classes from the App Store, and message us in the chat. Okay, so our representative very surely will connect you and add you free, add you in the free batches for the period of 1 week, right? And I'm requesting you, please don't call if you're not serious. Don't message if you're not serious. I'm only requesting only the serious aspirants to call or to message, for further details. Thank you all. Thank you so much.